So we take a muscle cell that's never seen your biochemistry and we add it to that muscle cell. Muscle is a good capture of your global metabolic health because we have a lot of it and it consumes a lot of energy. And so by using this muscle as the prototypical cell, we get a clean assessment of what's happening inside you and a good overall reflection of what's happening to your body in terms of health and disease. Mescreen then breaks it apart into multiple components. We want to know how your mitochondria are functioning and how their structure and dynamics is shifting in a unique kind of way. The first part of the test is a stress test. So we're putting your system against a stress test and seeing how your biochemistry adapts to that stress. The second part of the test looks at energy. How are you making energy? Are you using mitochondria? Are you using glycolysis? Is it efficient, inefficient? What is that energy state? And the two final parts look at the dysfunctional elements of mitochondria. Mitochondria are the largest source of reactive oxygen species in your body. We have mitochondria in every one of our cells except our red blood cells. And there's trillions of cells and there's trillions of mitochondria in those cells. And so you can imagine if they're the source of free radicals, this is where you get a lot of them built up. Free radicals cause damage to your proteins, lipids, membranes, and they create dysfunction. And so it's the thing that we want to be able to measure. So we've invented a way to look at how your environment either enhances or suppresses the expression of reactive oxygen species. The final component of the test is network dynamics. And so mitochondria are a living organism inside us. And like any living organism, they're social. They like to be connected to each other. Every chronic disease that you look at, one of the hallmark features is the mitochondria become isolated. They separate from each other. They can no longer sense stressors. They can no longer respond to toxins. They can't communicate with each other, so they can't sense and adapt to the environment. So we've developed a unique test where we can look to see where all of the mitochondria are in the cell. And then when your environment is adapted, we can see how that dynamic nature of your mitochondria either stays intact or starts to fall apart. So mescreen gives you a functional output for your mitochondrial aspects and the dynamics of mitochondria. Every other test that tries to do this gives you the potential of something that may or may not be there. Why not do a test that gives you the actual function of what your mitochondria are doing?